So, hey everyone, welcome to Simplified Explanation. Today I will be discussing about antigens of immunology. So, let's first understand what are antigens. Antigens are the substances which are capable of inducing specific immune response against that substance, or any substance or any molecule which gets recognized by the immunoglobulin receptor of B cell or T cell or T cell if it is complexed with MHC. So that substances or molecules are known as antigens. Now the antigens, they exist in various forms, and they can be in the form of chemicals, they can be in the form of toxins or proteins, polysaccharides, lipids, nucleic acids, which may be derived from microorganisms. So, those microorganisms can be anything like viruses, bacteria, protozoans, fungi, or any foreign substances. So that was all about antigens. Now let's talk about the major difference between the immunogenicity and antigenicity. So immunogenicity is basically the ability of a substance to induce a specific immune response, that is cellular and humoral immune response. So the substance which is capable of inducing the immune response is known as immunogen. Now our body or the biological system recognizes bacteria, viruses, fungi, and all microbes as the immunogens. So the most potent immunogens are the proteins, and then comes the polysaccharides. Whereas lipids and nucleic acids, they are not considered as the important immunogens until they get complexed with the proteins or polysaccharide. So all immunogenic substances are antigenic. I will be explaining this later. So next is antigenicity. So antigenicity, it's the property to be, or the ability to be specifically recognized by the antibodies generated as a result of immune response to given substance. Or we can say that any substance which is capable of binding with the final products of the immune response, such as antibodies or any kind of B cell receptor or T cell receptor, that substance shows the properties of antigenicity. So these do not produce immune response, they only get bind, and that is what is antigenicity. So not all the antigenic substances are immunogenic. That is, any substance which is having the property of immunogenicity will be having the property of antigenicity. But the reverse opposite is not true. It will not be true. For example, example of antigenicity is hapten. It is having the property of antigenicity, but it lacks the property of immunogenicity because it does not have the capability to induce an immune response. So it lacks the immunogenicity. Next, we will be talking about the factors influencing the immunogenicity. So, first factor is the properties of immunogens and the way these immunogens led to the activation of immune response, and the properties may be like foreignness, molecular size, chemical composition, heterogeneity, ability to be processed and presented with MHC molecule on the surface of the APC. Next can be the properties of the biological system of the host, because ultimately the biological system will determine whether the molecule which gets combined with the B cell receptor or the T cell receptor will induce an immune response or not. So the properties can be genotype of the recipient animal, immunogen dosage, and route of administration. So let's see them one by one, the properties of immunogen. So foreignness. So any substance or any molecule must be foreign or non-self to the biological system so that it can induce an immune response. Now the self and the non-self recognition, it is done during the lymphocyte development. Now what happens during lymphocyte development is that during that lymphocyte development, the immature lymphocytes, they are exposed to self components. And therefore these lymphocytes, they become tolerant to these self components. And that's how, so any antigen, if it is not getting exposed to the self component, that means that antigen will be considered as foreign substance, or it is considered as non-self. So the degree of the foreignness determines the degree of immunogenicity. The greater the phylogenetic distance between the species, the greater will be the structural disparities. For example, the BSA, it is not immunogenic to cow, but it is immunogenic to rabbit. So that was all about the foreignness. Now let's talk about the another property, that is the molecular size. So the active immunogen or active immunogen, they have the molecular size of 100,000 Daltons, while the poor and immunogens, they have molecular weight or the molecular size have a range of 5,000 to 10,000 Daltons. Next comes the chemical composition and heterogeneity, that is, if that molecule, if it is made up of single amino acid, then it will lack the immunogenicity, while if it is made up of different amino acid, then it will be having the property of immunogenicity. Next is the ability to be processed by antigen presenting cells. So if the molecule is large and insoluble, then it gets easily phagocytosed and presented by the MHC, and therefore it is much more immunogenic than the small soluble molecules. So that was all about the properties of immunogens. Next comes the properties of biological system. Now the genotype of the recipient, it determines the what type of immune response will be generated and to what degree the immune response will be generated. Next comes the immunogen dosage. Immunogen dosage means if single dose if it is given, then it will lack those immunogenic properties, but if multiple dose is given, then it will be having the properties of immunogenicity. Last one, the route of administration, that is there can be different ways of administration like intravenous, intradermal, intramuscular, subcutaneous, intraperitoneal. So these can be the ways like intravenous, into the veins, into the skin, into the muscle, into the peritoneal cavity, and this subcutaneous is beneath the skin. So this was all about the difference between the immunogenicity and antigenicity. Thank you.